The Atherton Tablelands, an area once covered by old-growth rainforest unique to this region of the world, have been almost entirely cleared for agricultural use and logging. The remaining fragments contain many of Australia's threatened species and continue to be degraded by forces such as climate change, invasive species, and anthropogenic impacts.
To combat the threat of habitat loss and degradation, local organizations and concerned citizens have made efforts to revegetate areas of cleared land around the tablelands. Dr. Noel Priest and Dr. Penny van Oosterzee bought Theaki in 2005 and began a large-scale revegetation experiment to determine cost and labor-efficient methods for optimum rainforest restoration. In our experiment, we compared low-diversity plantings with 6 tree species and high-diversity plantings with 20 forest tree species.
Dung beetles are a good indicator species as they are the first to suffer when a habitat is disturbed due to their drastic responses to changes in temperature, water, soil properties, vegetation, trophic resources, and parasitism. In addition, dung beetles are an essential mediator of many ecosystem services and functions, including secondary seed dispersal, bioturbation, fecal breakdown, increased plant growth, and parasite suppression.
Previous studies have shown that revegetated forests with more diverse tree species have greater variety in tree height and leaf size, which helps create a canopy that stabilizes temperature and humidity and increases leaf litter. They also have improved recruitment of new seedlings from animal seed dispersers, creating a rich understory. All of these factors create conditions suitable for a variety of dung beetle species that are more sensitive to their environment.
We conducted our research at Theaki and hypothesized that high-diversity plots would have greater species richness and greater functional group richness than low-diversity plots because they're more likely to have rainforest-like environmental conditions that create niches for many types of dung beetles.
We set pitfall traps in 8 revegetation plots, half of which were low-diversity plots and the other half which were high-diversity plots. We set 4 traps in each plot for a grand total of 32 traps.
To attract a wide variety of beetles, rotting mushroom bait was used for 16 of our traps and a mixture of navel dung for the other 16. Fresh dung was collected from a variety of native marsupials in the wild and from the Lumholtz Lodge Wildlife Rehabilitation Center.
For each trap, a hole was dug in the ground and a cup containing water and soap solution was placed in the hole. A metal mesh was secured on top of the cup to prevent large organisms or leaf litter from falling in. The bait was then suspended above the trap and secured with a twist tie. Finally, a plastic rain shield was installed at an angle in the ground uphill from the trap to prevent rainfall and runoff from flooding it.
4 days later, we returned to collect our traps. Several environmental measures were also recorded at Theaki, including canopy height and coverage, strata levels, the type of ground cover, and amount of woody debris across our transects.
Back at the lab, we began the process of sorting through our samples and separating out dung beetles from other insects. From there, we identified each individual beetle down to the species level, using identification keys created by previous researchers from the Queensland area. The most well-preserved beetles were then pinned and added to a reference collection intended for the use of future researchers.
We collected a total of 149 dung beetles across 19 species. We did not find a difference in the average number of dung beetles between the 2 plot treatments. However, high-diversity plots had a significantly greater average number of species present. We also tested the richness of functional groups in each plot type. Each beetle was classified by size, activity period, and behavior, so the functional group for one individual might be large nocturnal tunneler or medium diurnal roller.
The average number of these functional groups was also significantly greater in the high-diversity plots, confirming that not only do the high-diversity plots host more beetle species, but they also have a wider variety of ecological niches being filled by the beetles that are present. Based on previous literature, we thought that any differences in dung beetle community structures would be due to differences in environmental factors, but we found absolutely no statistical significance in any of the factors we measured.
Although many of our environmental factors indirectly affect light, moisture, and temperature, we did not measure these elements directly. It is possible that none of our environmental factors were significant on their own, but together they may have had subtle influences in high-diversity plots that made them more habitable for a greater variety of dung beetle species. In the future, we would want to measure light, soil, and moisture directly.
Although we were not able to find an environmental explanation for why the high-diversity plots had more species and functional groups, our results demonstrate the importance of using more than one approach to evaluate the success of reforestation schemes. Using species-level comparisons of dung beetles revealed subtle yet important differences in the ecosystem of each revegetation treatment. These differences were not detected by comparison of vegetation types alone. We hope that future researchers will consider dung beetles and other understudied taxonomic groups as viable ways to evaluate and understand restoration health.